Hello, everyone. Welcome back to my video tutorial for single-cell RNA sequencing data analysis. In this video tutorial, I'm going to continue my demonstration to show you how to analyze single-cell RNA sequencing data in Seurat v5. So in previous video, I showed you how to analyze just one single-cell RNA sequencing data in v5. So today I'm going to show you how to analyze more than one single scRNA sequencing dataset. This is where lots of people encountered problems, issues for using Seurat v5 at the moment. So I'm going to note the three different non-data sets generated by 10 times genomics techniques. So there will be no difference for the analysis process if you are analyzing dataset from three different repeats in the same experiment or dataset generated by different technologies. You can use the same code I showed you here for the analysis. So first we can note the three different datasets. The first dataset is the GSE 132771. We used this dataset for previous video. We can note the data in. You can see here is the large DGC matrix data for this dataset. Then we can create a Seurat object to keep genes that express the listed in 3 cells and keep cells that have at least 200 gene expression, and we set the project as GSE 132771. Then we can calculate the mitochondria DNA percentages. So now we can run the wiring plot. You can see we generated 3 wiring plot. We can select the parameters for the N features below 4,000, N count below 20,000, and mitochondrial DNA below 10% to subset the data. Now we can have a look at the metadata. You can see we have the barcodes as the row names. We have the origin ident as the GSE 132771. Then we have the N count RNA, N feature RNA, and percentage mitochondrial DNA for this dataset. So as I said, I'm going to show you the analysis in Seurat v5. We can have the C type for this dataset. If we run, you can see the RNA is a C5. And we can have a look at the cell numbers and the gene numbers in this dataset. You can see we have 3,616 cells, 17,574 genes in this dataset. So next we can do the same to note another dataset, GSE 128033. You can see here we noted the large DGC matrix data for this dataset. Then we can use the same parameter to create the Seurat object. Then we calculate the mitochondrial DNA percentage for this dataset. And we run wiring plot again. You can see from the wiring plot, we can keep the cells for N features below 4,500, N count below 20,000, and the mitochondrial DNA percentage below 10%. So we can subset the data. We can have a look at the metadata again. You can see it looks the same. Barcode, origin ident, N count, N features, and the percentage of mitochondrial DNA. Again, we can confirm the C class. You can see it is a C5 dataset. And we can have a look at the cell numbers and the gene numbers. You can see in this data set, we have 1,184 cells, 14,349 genes. Now we can load the last data set, GSE 128169. You can see here we loaded the large DGC matrix data. Then we can create a Seurat object, use the theme parameter. Let's run. Then we can calculate the percentage of mitochondria DNA again. Now we run variant plot. You can see from the variant plots, we can keep cells with N features RNA below 5,000, N count below 20,000, and mitochondria DNA percentage below 20%. Let's subset this dataset. If we check the metadata, let's run. You can see it looks the same. Barcodes, origin ident, N count RNA, N feature RNA, and percentage mitochondria DNA. And also we can confirm the assay class. You can see it is assay 5. We can have a look at how many cells and how many genes in this dataset. You can see we have 5,019 cells, 17,933 genes in this dataset. So now we loaded 3 Seurat objects. Then we can merge them into one object for integrated analysis. We can merge them and create a new Seurat object, named the object. Let's merge the dataset. Then we can have a look at the metadata for this object. Let's run. You can see again we have the barcodes as the row names. We have the origin ident as the GSE dataset name. We should have 3 dataset here. Then we have the N count RNA, N feature RNA, and percentage mitochondria DNA. Again, we can confirm the assay type. Let's run. You can see for this merged object, it is assay 5. So we created the merged object. We can remove the other 3 Seurat objects. So now we want to see the difference for the data structure in Seurat V5. If we click the object, you can see in the scRNA, we should have 3 different layers. If we click the RNA, you can see the layers. We click it again. You can see now we have 3 different layers. The count for GSE 128033, the count for GSE 128169, and the count for GSE 132771. If you use Seurat V4, here we only have 1 layer for the count. This is the difference in Seurat V5 because Seurat V5 was designed to do better data integration. It is more convenient to run different data integration methods. Before in Seurat V4, because the layers are in one layer, then we need to split the dataset into different layers. So here, because the V5 save the data in different layers, we don't need to split it again. So now we can run the analysis. First, we can normalize the data. You can see here, it normalizes the data for each layer. Then next, we can find the variable features. Again, you can see it goes through 3 different layers and find the variable features. But the analysis will identify the same set of variable features for all 3 datasets. We can have a look at the variable features. Let's run the plot. You can see we generated the variable features plot. We have 2000 variable features for all 3 datasets. Now we can scale the data. Because we normalize the data and also scale the data, we can have a look at the data slot again. We connect the object in the assays, RNA, and the layers. Now you can see we have 3 layers for original count and 3 layers for each dataset after normalization and 1 layer for the scaled data. Again, you can see Seurat wave saved the normalized data in different layers. So now we can continue the analysis. Next, we run PCA. For Seurat wave data integration, you have to run PCA first, because later, when we run different data integration method, we need the origin reduction method as PCA. So let's run PCA now. Then we can use the elbow plot to identify the best PC numbers for downstream analysis. Let's run. You can see we generated the elbow plot, and the point of elbow is somewhere here. So I would say if you choose PC number after 20 will be okay for the analysis. So now we can perform data integration for 3 different datasets in Seurat WAV file. We can have a look at the online tutorial for the Seurat WAV file data integration. You can see there are different methods you can use. For example, the CCA integration, RPCA integration, harmony, fast MNN. And also you can use the scVI method, because this method is a Python package. If you want to run in R, then you need to install the Conda environment in your R. So for today's demonstration, I'm going to show you the RPCA method and the harmony method for this data analysis. So first we can run the RPCA method. You can see in Seurat WAV file there's a new function called integrate layers, because 3 different datasets was saved in Seurat WAV file as different layers. Here we just need to set the method as RPCA integration. Then here you can see we need the origin reduction method, PCA. That's why we need to run PCA first. Then we can set the new reduction method as RPCA. Now we can run the data integration. Okay, you can see the integration process is very quick. Now we can find the neighbors, but now we set the reduction method as RPCA, so we can use PC numbers from number 1 to 30 to find the neighbors. Let's run. After that, we can find the cell clusters. I'm using resolution 0.8. And we can name the cell cluster name as RPCA clusters. So now we can run UMap. The reduction method will be RPCA. Then we can set the reduction name for UMap as UMap RPCA. Let's run UMap now. So we finish the analysis. We can have a look at the metadata. Let's run. You can see now, after the data integration, we have the RPCA clusters, and we also have the Seurat clusters. The cluster number are the same for each cell. We can close all the windows for the metadata. Now we can have a look at the Seurat object structure again. You can see in the reduction, we run the PCA, we also run the RPCA integration. Then we run the UMAP, and we named the UMAP as the UMAP RPCA. Then we can use the dim plot to look at the cell clusters. Let's go back. You can see now we can run dim plot. The reduction for UMAP will be UMAP RPCA. And we can group the cell clusters as RPCA clusters. Let's run. You can see we have 24 cell clusters here. If we group the cells by the origin ident, that's a different data set name. If we run, you can see now the cells were labeled by different data set. CSC 128.033 in red, 128.169 in green, and 132.771 in blue. You can see the cells from 3 different datasets were integrated very well. We can also use the split by function to look at the cell clusters. Let's run. Then you can visualize the cells in each cell cluster from different datasets. So I showed you how to integrate 3 datasets using the RPCA integration method. The advantage using Seurat V5, you can use different methods to integrate the dataset and save all the integration results in the same object. So now we can continue use the same object and perform the harmony integration method. You can see we can just use the same object. We already did the integration for 3 datasets using the RPCA method. Now we just set the method as the harmony integration, the origin reduction method still PCA, and we give a new reduction name as harmony. Let's run the harmony integration method for this dataset. Again, you can see very quickly we finished the harmony method integration. Now we can find the neighbors. We set the reduction as harmony for the harmony integration method. Then we find the clusters. We can set the same resolution as 0.8 and give a cluster name as harmony clusters. So you can see, use the same resolution with the harmony method. We only have 19 cell clusters here. Then we can run UMAP. And we give a reduction name for the UMAP as UMAP harmony. So after UMAP, we can have a look at the metadata now. You can see in the metadata, we run the RPCA integration before. We have the cell clusters number for the RPCA clusters. and we run the harmony integration method again for this dataset. You can see we have the cluster numbers for each cell after harmony integration. Because we just run the harmony integration, now the Seurat cluster number will be the same as the harmony cluster number for each cell. If we have a look at the object structure again, you can see in the reduction, we have the original PCA, we run the RPCA integration, we have the UMAP RPCA, then we run the harmony integration, then we have the UMAP for harmony integration. So now you can see the advantage using Seurat v5 for data integration. You can run more methods here. I just demonstrated two methods, RPCA and harmony for this dataset. So we can go back to look at the cell clusters from the harmony integration. We can use the dim plot. We need to set the reduction as UMAP harmony. Then we can group the cells by harmony cell clusters. Let's run. You can see on the UMAP, the name will be UMAP harmony. We have 19 cell clusters here. Then we can group the cells by their origin ident. That's the GSE data name for 3 different datasets. If we run, you can see the UMAP. Again, the cells from 3 different datasets are integrated very well in different cell clusters. If you want, you can still use the split by function to look at the cell clusters from different datasets. Let's run. So here you can see the cells from different datasets after harmony integration. You can see the UMAP is UMAP harmony. So now, because we have two different UMAP RPCA and UMAP harmony, we can run the feature plot for the classic mark genes for non-cell types on different UMAP. For example, here we can set the reduction as UMAP RPCA to look at the features for collagen 1A2, EPCAM, CLDN5, and PTPRC. Let's run. We can zoom in. You can see here we show the mark genes on the UMAP RPCA cell clusters. If we set the reduction as the UMAP harmony, let's run it again and zoom in. You can see now we show the gene expression on the cell clusters using harmony UMAP. I should use the cluster number name for each cells. For the Seurat clusters are the same as UMAP harmony analysis because we run the harmony integration after the RPCA analysis. Let's use EPCAM as an example to run the wiring plot. You can see now we only have 19 cell clusters here. So wiring plot uses the Seurat clusters as the default setting. Then here you can see the total number of cell clusters are the same as the harmony integration. So far it is okay, you can run feature plot and wiring plot. But if you try to use the find the marks function to identify mark genes for each cell cluster, because the Seurat v5 save the data as layers, then you cannot run the find the markers function. Let's try it. You can see we have an error in find the markers function. It suggests you to run the join layers function. This is where many people encounter problems. So let's have a look at the data structure again. Before we run the joint layers function, we can connect the object in the assays, RNA, and the layers. You can see we have 3 count layers for each dataset and 3 data layers for each dataset. They are normalized dataset. So now we can run the join layers function for the object. Let's run. We can have the layers now for the RNA. If we run, you can see we only have data, count, and scale the data layers now. Let's connect the object and have a look at the data structure again. You can see in the assays, RNA layers. Now we only have the data layer and the count layer. So we don't have the layers for each data set. So now if you run the find the markers function, for example, we can identify the mark genes for cluster 1. Let's run this function again. You can see now you can use the function to identify mark genes for different cell clusters after join the layers. Okay, I'm going to stop from here for today's demonstration. You can see we can easily use the Seurat V5 to do data integration. Because the Seurat V5 save the data in layers, you will encounter some problems if you just load your data from previous analysis in Seurat V4, or you analyze the data in V5, you need to join the layers for other analysis. So after join the layers, the data structure looks similar as V4 data structure. At the moment, lots of other packages were not updated to use the Seurat object analyzed by the V5 version. So I hope this video could help you understand better the data structure in Seurat V5 version. Then you know why there are errors or issues when you run the analysis.